and just an acknowledgement of all of our sponsors and partners there. Without all of these partners and sponsors, this event would not be able to run. So it's an absolutely amazing day yesterday, and today will be no different. And with that, I'd like to introduce Dr. Emilia Martin. She's currently a project manager for the Australian Network for Plant Conservation and has coordinated updates for the third edition of the Plant Germplasm Conservation in Australia. And I think that's a bit like herding cats, so I think Emilia has done an amazing job. Emilia has a research background investigating seed lifespan and germination with a focus on improving seed quality for conservation, storage of Australian native plant species. She has also a growing interest in science communication. Emilia's research is based at the Australian Plant Bank, part of the Australian Institute of Botanical Science. When she's not at work, Emilia is usually looking after her time-intensive germination experiments, namely her 3 kids, or reading a good book. Over to you, Emilia.
Thank you, Sal. I'm proud and humbled today to join you for the launch of the 3rd edition of Plant Germplasm Conservation in Australia. When Professor Leslie Hughes launched the previous edition in Melbourne in 2009, she noted that these guidelines were a message of hope, and I've looked at her inscription many times while we've been working on this new edition. I'd like to suggest that this 3rd edition is a message of hope and action as we come to terms with the urgent need to conserve plant diversity, while the threats are overlapping and increasing in magnitude. As humans, we desperately need to see hope combined with action. We need to look out for projects, initiatives, and individuals who are working so hard to conserve plant diversity, habitats, and threatened species, and to use ecological restoration and translocation to win back landscapes. The Australian Network for Plant Conservation is proud to have played a part in sharing knowledge and connecting people and organisations dedicated to conservation of Australia's native plant species over the last 30 years. Production of this handbook for ex situ plant conservation has been an incredible team effort. The lead authors, co-authors, case study contributors and steering committee have revised 8 chapters and drafted 7 new chapters over the last 18 months. I'd particularly like to thank the lead authors, Kathy, Marlene, Anne, Dave, Karen, Meg, Bryn, Amanda, Nushka, Tom, Danny, Graham, and Andrew. Their co-authors each brought something important to the table, and each writing team was responsive and persistent through the revision process. The first 6 chapters provide a step-by-step guide to germplasm conservation, starting with planning, followed by genetic guidelines, collecting strategies, seed storage, and germination. The next 4 chapters discuss techniques that may complement seed storage or be the only mechanism of exceptional species, such as tissue culture, cryopreservation, nursery practice, and living collections. The following 2 chapters outline the types of germplasm collections suitable for species with special requirements, such as orchids, fungal symbionts, ferns, carnivorous, and parasitic plants. The final 2 chapters provide practical advice for managing risk within collections and strategies for responding to crisis situations, as well as guidelines for the maintenance of use of all types of germplasm collections and associated data. Please show the chapter authors and co-authors your appreciation in the discussion. This edition includes an incredible 50 case studies that highlight the aims, processes, and partnerships that underpin ex situ plant conservation. I encourage you to read every one, to learn from what these authors are sharing, and let yourself be inspired by their actions. Whether they are encouraging volunteers to get involved, refining techniques for breaking dormancy, bulking up collections through seed production, or using collections for translocation, there is so much we can do to make the best use of every germplasm collection. Please show the case study authors your appreciation in the discussion. We have dedicated this edition with gratitude to all those who introduced us to the beauty and complexity of Australia's flora. We owe a huge debt of gratitude to our families, friends, and colleagues, past and present. They are also dedicated to the children and grandchildren we know, who inspire our efforts to conserve Australia's biodiversity. The last 2 years have reinforced that the relationship we have with the natural world sustains us in so many ways, and the relationships we have with each other make life meaningful. On that note, I would particularly like to thank the Germplasm Guidelines Steering Committee for their support. Joe Lynch, Lucy Commander, Kathy Offord, Bob Makinson, David Coates, Linda Broadhurst, Damien Wrigley, Sal Norton, Kath Whitehouse, Lydia Guza, Neville Walsh, David Bush, Rebecca Stanley and Tony Ord. I also owe a huge thank you to my colleagues at the Australian Institute of Botanical Science. It is a pleasure and an inspiration to work with people who love plants so much. This work would not be possible without the support of our generous funding partner, the Ian Potter Foundation, and our project manager there, Louise Arkles. Their funding allowed for project coordination based on attainable and flexible goals. Importantly, it also means these guidelines and accompanying videos are beautifully presented and available for free download, which is critical for accessible and open sharing of this valuable information. I'd like to introduce the CEO of the Ian Potter Foundation, Craig Connelly, to say a few words ahead of the official launch. Craig commenced as CEO of the Ian Potter Foundation in 2015. He spent 25 years working in a variety of roles in the Australian financial services sector and is now dedicated to contributing to the community sector. Craig is an experienced business analyst and successful manager and has been an effective mentor to many young professionals throughout his business career. Craig has previously been an external member of the Advisory Committee for the Sustainable Farms Initiative at ANU and a member of the Natural Capital Expert Advisory Group. He's founding trustee of the National Parks Conservation Trust and currently a director of Water Trust Australia and SANE Australia. Thank you for joining us today, Craig.
To you and good morning, everybody attending from wherever you are located around Australia this morning and internationally. I'd also like to acknowledge that I'm coming to you today from the traditional lands of the Wurundjeri people of the Kulin Nation, and I pay my respects to elders past, present and emerging. The Ian Potter Foundation is very pleased to have supported the Australian Network for Plant Conservation with a $200,000 grant to comprehensively update their flagship publication, Plant Germplasm Conservation in Australia, Strategies and Guidelines for Developing, Managing and Utilising Ex Situ Collections. That's a mouthful. In preparing for this session, I chatted with Emilia early last week, and throughout that conversation, it became obvious that there are layers of positive outcomes that have flowed from this relatively straightforward and modest grant from Potter's perspective. It's clear in Emilia's opening comments that the steering committee has been incredibly influential in overseeing the update of the guidelines. They've leveraged significant in-kind and expert support from eminent individuals and organisations who've all contributed immensely to the content review and update of the guidelines and have done so pro bono. The fact that such support, coupled with the efforts of members of the Australian Network for Plant Conservation, means there's a strong scientific basis for all updates to the guidelines as now published. That's critical to Potter. We've been a strong supporter of scientific endeavour since the foundation was established in 1964. The funding also supported efforts by the Secretariat to engage leading scientists at different stages of their careers and from a geographically dispersed and diverse area to contribute to the update in an inclusive and engaged way. And pleasingly for IPF, and as Emilia has already articulated, the dissemination of the updated guidelines is going to be to a very wide audience and is free. And possibly an unintended consequence of the funding that's been provided has been to facilitate two-way exchange of material with international contributors, effectively raising awareness of the updated guidelines and also facilitating connections that might foster future collaborations. As a funder that's supportive of scientific endeavour, this project ticked a lot of boxes for Potter. The work ensures that the guidelines remain relevant, they're up to date, they're available to as wide and an engaged audience as possible, and they're based on world-class science. And a small budget has also been set aside to evaluate the project, and I look forward to discovering the evaluators' conclusions in years ahead. It's now my pleasure to introduce our next speaker, Dr. Kathy Offord. Dr. Offord is the Manager of Germplasm Conservation and Horticultural Research for the Royal Botanic Gardens and Domain Trust. She is based at the Australian Plant Bank, part of the Australian Institute of Botanical Science, in southwest Sydney. Dr. Offord is also an adjunct professor at the School of Biological, Earth and Environmental Sciences at the University of New South Wales. Specialising in the conservation and horticultural development of Australian plants, Kathy's work encompasses seed biology, tissue culture, cultivation and translocation of threatened species. Kathy was the lead editor and author of several chapters of the previous edition of Germplasm Guidelines and the chair of the steering committee for the revision and publication of the third edition. Welcome, Kathy.
On behalf of the steering committee for the third edition of the Australian Germplasm Conservation Guidelines, I want to sincerely thank all of the contributors to this and to the two previous versions. As I reflect on my own career as a conservation scientist, I can measure the real-time increase in ex situ conservation science by the size and the scope of the successive guidelines that have been produced. The first guidelines were published by ANPC in 1997. These were groundbreaking guidelines and they ran 40 pages, which were overseen by the editors Darren Tushel, Mark Richardson and Kingsley Dixon. This document became the wayfinding document for those of us who were grappling with the burgeoning needs that emerged in the 1990s to ramp up scientific efforts to conserve plants ex situ and in situ. 12 years later, in 2009, Trish Marr and I edited the second edition, and the scope and the magnitude of the information that was available had increased to the edition to 190 pages, including 19 authors. Fast forwarding another 12 years brings us to this 2021 revision. This is a compendium of great science and practical wisdom. It reflects the vastly expanded activity in science and practice of ex situ conservation over the past 12 years. In all, there are 15 chapters and an astounding 50 case studies written by 78 authors. Added 5 reviewers, and there are more than 330 infographics, illustrations or graphs. The printed document itself will run to an impressive 500 pages. This latest version has been overseen by the extraordinary Emilia Martin, who is herself a researcher and contributor to Australian plant science and science communication over many years. Emilia has wrangled the multitude of authors and reviewers with gracious tenacity, and it's due to her efforts that we are launching these guidelines on time and on budget today. I also want to mention the incredible Siobhan Duffy, who's made these guidelines a pleasure to read with her amazing design and editing talents. The constant in the years between the successive versions of the guidelines is the never-failing support of the Australian Network for Plant Conservation, who, despite often lean times, have always been there to ensure collaboration between scientists and conservation practitioners, and to provide workshops, conferences, guidelines, and other products to enable active plant conservation. Thank you, Joe Lynch and the team at ANPC for providing the knowledge bridge between science and effective conservation policy and action. I must also mention the support of the Australian Seed Bank Partnership, the powerhouse of Australian seed conservation. Thank you to Damien Wrigley and to all of the other seed bank partners. I'm proud to have been involved with the development of these guidelines, and I'd like to acknowledge the wonderful support and thoughtful guidance of fellow members of the steering committee. Special thanks goes to Linda Broadhurst for heading up the application for funding of these guidelines, which has been provided by the Ian Potter Foundation, who have been so supportive in our collective efforts to and it is now my great pleasure to introduce Professor Tim Entwistle to officially launch the third edition of the Australian Germplasm. He's a highly respected scientist, scientific communicator and Botanic Gardens director. Tim is currently the Director and Chief Executive of the Royal Botanic Gardens in Director of the Royal Botanic Gardens and Domain Trust in Sydney. Tim is an expert in freshwater algae, but he has a broad interest in plants. Tim.
you very much, Kath, and it's great to be here today and lovely to be invited. I'd first of all like to acknowledge that I'm speaking also from Wurundjeri land and pay my respects to elders past and present, particularly any elders who might be with us here today. This is an amazing occasion, and I was thinking when I was preparing for this today that we're often very apologetic about seed banking and in fact about ex situ conservation in general. I suppose we're very aware that recreating nature from a few packets of seed is close to an impossible task. That's kind of not really what it's about. But the fact that we're already making withdrawals from our germplasm banks for restoration, relocation, responding to bushfires, climate change, that relentless land clearing, demonstrates, as if we needed it, that the situation is quite dire for our native biodiversity and how important things like ex situ conservation are already and are becoming so. So while we know its limitations, seed banking is important, but not only that, it's quite romantic and very uplifting for many. And I don't think we should lose sight of that. I think everyone has collected or stored or tried to germinate seed, and that can be a tomato plant, it could be an avocado, or it might be a local gum tree. And seeds are often metaphors in songs and writing and used for hope and renewal, you know, from little things, big things grow, and many such lines. And for those of you who might be wondering about the bad seeds, apparently that band name comes from a 1956 horror movie, so it's entirely unrelated to all this. But look, seeds are fascinating, and old seeds are even more fascinating. 16 years ago, I was thinking, when I was freshly appointed as director at Sydney's Botanic Gardens, and there was a media report came out about successful germination of a 2,000-year-old date palm. Now, very few seeds last more than a couple of hundred years, and even that seems a little bit excessive in terms of evolutionary advantage, but there you go. When I was preparing to talk about the old palm seed on Sydney radio at the time, I, of course, chatted to Kathy, who's just introduced me, and did a bit of Google research, as you do. And people repeat stories about wheat seed surviving in the pyramids and Egyptian tombs, but these are mostly carbonised, non-viable seed that they might look okay, but they're dead, effectively. There's records of 10,000-year-old or perhaps older lupin seeds found in Arctic lemming burrows. These are also dead. And in fact, I've got an acorn on my bedroom windowsill at the moment that's 12 months old, and that's probably dead as well. I only found out recently that acorns only live for about 6 months or so, even in squirrel hollows. So I'm going to just keep that there for beauty purposes only on my windowsill. But with the right care and condition, seeds do last for a long time. And prior to that 2,000-year-old date palm seed, the oldest known seeds to germinate were lotus seeds, I think, a Nelumbo from a lake bed in China. These were said to be 1,288 years old, so very old. And they did germinate, but didn't quite make it into fully grown plants, which I do remember is something that Kathy was explaining to me, that seeds can be alive, but just alive, not quite enough. And there are successful seedlings that have been grown from 400, 450-year-old lotus seeds, although they were still a little bit wonky too, I think. But, you know, when you get into the hundreds, there are convincing stories, and there are, you know, there's a story of a 600-year-old seed from a ceremonial rattle in Argentina. Q always tells its story about the 200-year-old leucospermum seed from Africa, which was stored in a leather wallet and could be, you know, regenerated and grown into a plant. In Sydney, we had our own tale from 1890 when there's 100-year-old golden wattle seeds that have been stored in the belongings of someone's deceased mother. And they were germinated, and 8 out of 10 of those survived 100 years. So these are great sort of headline-grabbing stories, but they're not really, as everyone here would know, what seed banking is really about. A couple of wheat, lupin, lotus or date seeds is not going to protect the world from rampant land clearing, extreme bushfires, pests, disease attack, not to mention climate change. So that requires something altogether more sophisticated than a wallet or a dead person's chattels or even a pyramid, let me say. We need the infrastructure, we need the seed banks, the nurseries, we need the knowledge, and we need that know-how about what we're doing, which is why I'm delighted to be here today to help launch these guidelines, partly because I do enjoy a good story about old seeds, but I'm also aware that seed banking is much more than that and quite complicated and complex. Now, I know that because I'm, as you heard, head of Botanic Garden here in Victoria, which is one of Australia's 10 conservation seed banks, as well as we have here a flourishing orchid conservation program. We have a spore bank about to be created, and a recently expanded ex situ living collection program in our nurseries too. I was previously head of the Botanic Gardens in Sydney when Plant Bank was just a twinkle in a few people's eyes. We had a promise of a few $1 million to build it, and the money came a year after, I think, the launch of the second edition of these guidelines in 2009, which was also, interestingly, the year we sent some packets of seed to the space station with an astronaut, Greg Chemitov. Speaking of stories that add to romance more than science. But anyway, it was a fun time back there in 2009. And then after that, also, I spent some time at Kew Gardens for a few years looking after conservation living collections and estates there, which gave me responsibility for the wonderful Millennium Seed Bank. And that, of course, is a founding partner for so many seed banks around the world. I'm also delighted because I'm a member of the Council of Heads of Australian Botanic Gardens. As you've been hearing, they're governing or looking after the Seed Bank Partnership in Australia, and which is our premier germplasm network, of course. And working in a botanic garden does give you a particular perspective on all this. And I'm not going to talk today about the value of preserved plants in our herbaria, the learning programs, the activities that we do to encourage changes in behaviour, or about scientific research that goes along with those preserved and living collections. There's a whole lot of things that botanic gardens do, of course, that contribute to conservation, contribute to fighting climate change, and are associated with seed banking. But I did want to say a few words about what I like to call conservation horticulture. If seed banking is not just about old seeds or sending seeds into space, what is it about? Well, I think it's about horticulture in its broader sense of that term. And I've been fond of saying recently that gardening is the answer to our plant biodiversity crisis. It's an oversimplification, but I do think that gardening is going to be a key part of what we have to do with the kind of impacts that are happening, particularly with climate change, but also big things like bushfires that are driven by climate change. Now, conservation gardening, or more formally conservation horticulture, also used to be a bit of a fringe science, perhaps a little bit like seed banking, and it was lumped in as something at the side of kind of where real conservation took place. And as I said at the very start, we now know that's not enough. You know, we need to draw on our seed banks now, and in a crisis such as our response to climate change, we do need to base our action on the very best scientific evidence and very best advice. And that all points, I think, to a very well managed ex situ plant collection. Now, none of us want a kind of a parallel universe of living dead plants in nurseries and fridges while the plant steams and burns and drowns. That's not the outcome we want. But just as we accept hospitals, care units, medical intervention for humans, particularly at this time during the pandemic, we have to embrace, I think, ex situ conservation in that same vein. While the analogy with medicine does have a few weaknesses, another strong parallel for me is that these ex situ conservations are partly insurance, but they're also for research and other purposes as well. So it's for healing now, as well as gathering the knowledge we need to heal later. What I like to call a plant rescue and care unit, which is like an intensive care unit in a hospital, is now really part of our business here at Royal Botanic Gardens Victoria, and part, I know, of the business of many of you here today. So I was very pleased to see in this new guidelines a section on propagation and nurseries being pumped up a little bit. So some of the chapters here have been really added to since the last editions. And as with so much of the book, drawing on expertise from all around the country. I think that's a real strength of these guidelines, the many voices, the many areas of expertise, and the fact they draw it all in from all those different places. Now, the problem we are addressing is, of course, a rather big problem. There's 330,000 known plant species alive today that produce seeds, or named plants. We do know there's a lot of seed banks in the world. It's been calculated there's 1,750 of them in the world with about 6 million accessions, but, and the but is, most of these hold crop plants and wild relatives. So it's the conservation seed banks we're really interested in here today. There are about 50,000 species or taxa in conservation-based seed banks. And of course, thanks to orchids and things like that, there's millions of seeds as well. But 50,000 species in those seed banks, possibly more. And that means that we've got at least 15% of what we know of the world's seed-bearing plants already stored in seed banks. That's, you know, that's a really positive achievement, I think, actually, to be on the way to holding that many seeds, and probably well beyond that now. Importantly, of course, many of those with a single accession or collection, as again, it comes out through these guidelines, it's critical to have more than that. Here in Australia, with about 23,000 vascular plant species and 84% of them not growing anywhere else on the planet. So this is an amazing flora we're looking after. If I interpret that, what I read correctly in the guidelines, our Australian seed banks hold perhaps 10,000 of those, and that's in about 20,000 separate accessions, with most of them, I should say, duplicated in the Millennium Seed Bank queue. So we may have nearly half of our known species, Australian plant species, in seed banks, although I do know that figure's a little bit rubbery. I know Damien is working on getting a real estimate very soon on that. But again, a handful of seeds for each species, and on average, perhaps it's about 2 collections for each. So there's a little bit of work to do there, but we are starting to get that material into the bank, ready to pull out when we need it for whatever purpose. Our focus at the moment, though, of course, is on rare and threatened species, and about 10% of Australian plant species are threatened with extinction, or we know are threatened with extinction, and many more are rare, and I would suggest on the edge of listing as well. So what we do know more precisely, and you can read this in the guidelines, is that we have close to 68% of federally listed species, that's about 930 of them, in our seed banks. Now, that may drop a tiny bit, or who knows how much, as we discover what we may have lost after the recent bushfires, and of course will lose through climate change. So that number of endangered and threatened species is likely to increase, and we need to keep continuing to get more of those into the seed banks. Then, of course, there's bryophytes, there's fungi, there's algae, there's other things we might want to also store as well. So there is plenty of work to do, and plenty of work to do properly. But as Emilia has said today, this guide should definitely be seen as a message of hope. We can do something constructive to save plants, and this tells us how we can do that. It's evidence-based, it's science-based, it's best practice with 50 constructive case studies on how to care for seeds, for plant tissues and whole plants off-site and look after them when they're not in nature. I do sometimes get the feeling when I talk to people outside this virtual room that seed banking is like shoving a bag of seed into an old cupboard, or or a pyramid, or preferably most people think under a few meters of permafrost, but then shutting that door and walking away. And then suddenly you can magic back the habitat by going in, pulling the seed out and popping it back in the ground. Now, anyone assisting with the incredible community restoration, revegetation projects that are going on, anyone associated with seed banking, germplasm banking, will know that is not the case. It's a rather difficult thing to do and requires a lot of expertise and a lot of experience. And the difficulty and the challenge starts with the collecting and storage of that germplasm in the first place, which is why this collection of very wise words and experience from over 78 experts in the field is so important. There's really a lot in here. It's 500 pages and written by so many different authors and experts. There's advice on collecting, on storing and growing plants, as you've heard, and then to be used for translocation, reintroduction and rewilding, or for research or public programs or other particular uses. It answers those kind of immediate questions. What do you put in your ex situ collection? How much of it? For how long? which bits are the plants, and through detail on different storage techniques, and right through to breaking dormancy. And in this new edition particularly, there's even more information, as you'd expect, on genetic integrity, on difficult to store and grow plants. There's more on orchids, of course, special collections like carnivorous and parasitic plants, on spores, pollen, risk management, a whole lot more. So lots and lots of information. What does appeal to me, though, is the mix of practical and theoretical. So you can dip into this guide to whatever level you think is appropriate. And it ranges from beautiful graphics and lovely summaries through to tantalizing mathematical formulae and quite complicated experimental results. But it's really a little bit there, a little something for everyone. We're told that there's 10 conservation seed banks in Australia, including the one here in Melbourne, and holding, as I said, 68% already of our threatened flora. And I think in a positive vein too, 92% of Australian plant species are relatively easy to store in one of those banks. That is, they're not recalcitrant or non-orthodox, non-orthodox. Of course, as the book says, we want more genetic diversity in there. We want more plants, and we want more of those things that we haven't quite got into those banks already. But it is really good news. So I today want to finish by thanking everyone who's been involved in this production, congratulating everyone, to the authors, to the experts involved in this edition, and to those who were involved in the first two as well, because it does build on their shoulders. It really is a collaborative effort, and I can't imagine the coordinating and cajoling needed to bring it to completion. I gather from Emilia it's a bit of a COVID project, and amazing to do it during the last couple of years and get it to completion. So well done to all the authors, led by Emilia, Kathy, Trish, and the others that have been mentioned already. Friends, colleagues, and wonderful people. So they've done a fantastic job. So well done, you guys. And finally, I would like to thank also Ian Potter Foundation for showing their support. It's really good to see them supporting this project, and the Australian Network for Plant Conservation for their persistence and dedication to this cause. Now, just before I do the official launch, and I have to work out how to do this with a virtual publication, I'll think of a way. We're going to show and share with you a short video now that hopefully doesn't say all that I've said, but it will tell you a little bit more and in a far prettier way. Thank you.
From deserts to wetlands, rainforest, woodland, and alpine ecosystems, Australia's flora has evolved to survive diverse climates and stresses.
We have more than 23,000 plant species.
90% of the plants in Australia are only found in Australia. They're endemic to Australia.
24 years ago, the Germplasm Guidelines were developed. These guidelines bring together our expertise in preserving Australia's unique flora and their genetic material away from their natural habitats. But in the same 24 years since these guidelines were developed, Australia's flora has faced increasingly complex challenges.
Australia has one of the worst global extinction records. The estimates of species extinction are between about 55 and 70 plant species, and more than 10% are still threatened with extinction. This is largely because of many co-occurring threats, including weeds, pests and diseases, overgrazing, habitat loss, changed fire regimes, and of course, climate change.
Some of the threats are so big, like a warming climate and the uncomfortable nature of how that feels. And the idea of having frequent severe wildfires covering most of eastern and western Australia, that's a serious issue for everybody who lives in this country. So you've got to start to deal with these threats. You've got to start to deal with them soon.
Preserving populations in the wild is vital, but is inadequate to alone protect our flora. We must respond through improving our off-site or ex situ conservation methods, which is why the Germplasm Guidelines have been updated.
There are new guidelines on the genetics of capturing and maintaining an ex situ collection. There are also new guidelines for species that need extra care in ex situ conservation, so orchids, special collections, and non-orthodox species as well. There's a lot of new information on identifying and conserving species that just don't suit conventional seed banking.
The updated Germplasm Guidelines are designed with use in mind. Use by volunteers, researchers, students, community groups, not-for-profits and government. With over 68% of Australia's threatened plant species represented in conservation seed banks, the way we preserve populations off-site or ex situ than ever. Ex situ collections aim to safeguard species for use in the wild, either now or in a future we cannot predict.
Some very familiar faces and voices there. Well done again, Emilia. Well done, everyone. It gives me great pleasure to launch Plant Germplasm Conservation in Australia, Strategies and Guidelines for Developing, Managing and Utilising Ex Situ Collections, 3rd edition, timely, necessary and perfectly pitched. Brilliant job, well done. Thank you. I think it's time for applause and fireworks.